Hi everyone, welcome to my A-level biology lecture series. In this lesson, we will explore anaerobic respiration. At the end of this lesson, you will be able to outline respiration in anaerobic condition in mammals involving lactate fermentation and also in yeast cells, which is ethanol fermentation, as well as explain why the energy yield from respiration in aerobic respiration is much greater than the energy yield from respiration in anaerobic condition. Let's get started. So before we go into anaerobic respiration, just a quick recap how we can regenerate NAD. So NAD can be regenerated in two ways. First, through the electron transport chain and oxidative phosphorylation, but there is a term and condition. Oxygen must present and act as a final electron acceptor. So in the presence of O2, hydrogen atoms are transferred to electron transport chain by reduced NAD, and therefore NAD molecules are regenerated. However, in the absence of O2, anaerobic respiration occurs, which can help to regenerate NAD molecules. When oxygen is not present, hydrogen atom carried by NAD cannot be removed by combination with oxygen. So we know that in this case, electron transport chain stopped functioning, and there is no further ATP is synthesized by oxidative phosphorylation. So the reduced NAD cannot be reoxidized back to NAD, and the link reaction and Krebs cycle stop. So therefore, oxidative phosphorylation can't take place without oxygen. And in this case, how the cells actually gains ATP. So under anaerobic condition, the cell needs to continuously gain a net of 2 ATP molecules through glycolysis. To achieve this, hydrogen atom from reduced NAD must be transferred, okay? Because during glycolysis, we know that oxidations of triose phosphate to form pyruvate involve NAD. NAD must accept hydrogen atom from triose phosphate in order to allow glycolysis to continue. However, NAD has a limited concentration, okay? So it's not like unlimited supply. We need to regenerate back NAD. So therefore, under anaerobic condition, we must regenerate back NAD as well, not through oxidative phosphorylation, but through the anaerobic respiration. So this transfer the hydrogen atom occur through anaerobic respiration. So there are two different anaerobic pathways that can help to regenerate NAD through alcoholic fermentation in the yeast cells, as well as the lactic fermentation in mammalian cells. Let us look at alcoholic fermentation that takes place in the yeast cell. So we know that in the yeast cells, pyruvate is converted to ethanol in two steps, which occur in cytoplasm during alcoholic fermentation. As we know that through glycolysis, glucose is oxidized to form pyruvate. During this glycolysis process, NAD accepts hydrogen atom from triose phosphate to form reduced NAD. So this reaction allows the cells to harvest a net gain of 2 ATP per glucose molecule. However, in order to regenerate back NAD in anaerobic condition, the yeast cells have to continue, okay, or have to regenerate back NAD through anaerobic respiration, which is alcoholic fermentation. So in this alcohol fermentation process, first decarboxylation occurs, where you can see that 3 carbons pyruvate releases carbon dioxide and form 2 carbon ethanol, or also known as acetaldehyde. Okay, so you can see that acetaldehyde is 2 carbon compound. After decarboxylation, the ethanol is reduced by reduced NAD to form ethanol. So how this occurs? In the presence of the enzyme known as ethanol dehydrogenase, NAD, okay, is regenerated because reduced NAD donated hydrogen atom, and this hydrogen atom accepted by acetaldehyde or ethanol. Ethanol is reduced to form ethanol. At the same time, reduce NAD, because reduced NAD releases hydrogen atom, it itself is oxidized back to form NAD. Okay, so this reaction is catalyzed by an enzyme known as ethanol dehydrogenase. So alcohol fermentation enables the continued production of small yield of ATP in yeast cells, even when oxygen is unavailable to act as a final electron acceptor. So the whole idea of anaerobic respiration, okay, is to make sure that glycolysis continue. As long as glycolysis is continued, then we will be able to harvest or to gain a small yield of ATP, which is a net gain of 2 ATP per glucose molecule. In order to make sure that glycolysis reaction continues, we must regenerate back NAD. So alcoholic fermentation pathway, which involve decarboxylations and as well as the reductions of ethanol into ethanol, allows the regenerations of NAD, and with the present NAD, glycolysis can continue. However, the productions of alcohol, okay, or ethanol in this case, are toxic. So as a result, this reaction cannot continue indefinitely. So the ethanol produced in alcohol fermentation cannot be reversed. So unfortunately, the remaining chemical potential and energy in ethanol is wasted. Okay, unlike a human and some animal will be able to metabolize ethanol, but for yeast cells, they can't. So about 8 to 10% of the ethanol concentrations is high enough to kill the yeast cell. So whatever chemical potential energy in ethanol is wasted. So what are the applications of yeast in industrial process? First of all, humans have been used yeast in brewing, wine making, and baking for many or even thousands of years. And particularly in the baking process, the carbon dioxide produced during alcohol fermentation by baker yeast help the bread to rise and to be airy. So let us look at the second type of anaerobic respiration, which is a lactic acid fermentation, which occurs in mammalian mast cells. So human mast cells or mammalian mast cells synthesize ATP by lactic acid fermentation when oxygen is limited. Okay, so as usual, this diagram shows again the glycolysis process where a glucose molecule is oxidized to form 2 molecules of pyruvate, and in this process, NAD is involved. Okay, so NAD is reduced to form reduced NAD, and the glycolysis process allows us to harvest a net gain of 2 ATP per glucose molecule. However, to make sure that glycolysis process continue, we must regenerate back NAD. So in humans mast cells, lactic acid fermentation pathway allow us to regenerate back NAD. So during this process, pyruvate is reduced directly by the reduced NAD to form lactate. Okay, so this reaction is catalyzed by the enzyme lactate dehydrogenase. So basically, reduced NAD donates hydrogen atom to pyruvate. So pyruvate accepts hydrogen atom, and pyruvate is reduced to form lactate in a single step process. And because reduced NAD donated hydrogen atom, it itself is oxidized to form back NAD. With the presence of NAD, glycolysis can continue. Compared to alcoholic fermentation pathway, there's no release of CO2 during lactic acid fermentation. And as we know that the regeneration of NAD allows glycolysis to continue under anaerobic condition. So what is the effect of accumulation of lactic acid in muscle cell? So the accumulation of lactic acid due to the presence of protons definitely lowers the pH inside the muscle cell, making the environment now more acidic. So the acidic environment can interfere with enzyme function and disrupt the metabolic process, leading to muscle fatigue. As lactic acid builds up, efficiency of glycolysis decreases, limiting ATP production and stop muscle performance. However, this effect is usually temporary because lactic acid or lactate is eventually removed and converted back into the glucose in liver through a process known as Cori cycle when oxygen becomes available. In this diagram, you can see that the production of lactic acid, this lactic acid now can be removed from the muscle cells into the bloodstream, and through this blood circulation, the lactate is transported to the liver. Inside the liver, there are reactions to convert this lactate, back to glucose, or lactate can be oxidized to form CO2 and water, as long as oxygen becomes available. So the lactate pathway is known as, or can be said that it is a reversible reaction. So lactate is carried by the blood plasma to the liver and converted back to pyruvate. And the liver oxidizes some of this incoming lactate to carbon dioxide and water through aerobic respiration, provided that oxygen is available again. So the remainder of the lactate is converted by the liver to form glycogen. So the volume of oxygen required after exercise, as we know that after exercise, our breathing rate is still very high. We have extra consumption of oxygen after exercise. So this extra volume of O2 is required after exercise to remove lactate and also to prevent the toxic accumulation and to restore back the normal muscle function. So this volume of oxygen is referred as oxygen depth. So in our syllabus, we need to know the differences between alcoholic and lactate fermentation pathway. In terms of type of cells, yes, alcoholic fermentation occurs in the yeast. However, lactate fermentation occurs in mammalian muscle. Decarboxylation takes place in alcoholic fermentation where CO2 is produced. However, there's no carbon dioxide is produced through decarboxylation in lactate fermentation. The end product alcoholic fermentation is ethanol. For lactic fermentation, is lactic acid or lactate. The enzyme involved in the production of the end product alcoholic fermentation uses ethanol dehydrogenase, where ethanol is reduced to form ethanol. However, in lactic fermentation pathway, the pyruvate is reduced directly to form lactic acid by lactate dehydrogenase. So alcoholic fermentation has it's a two-step pathway. However, lactic fermentation is a single-step process. So since alcoholic fermentation is a two-step process, there is an intermediate compound known as ethanol or acetaldehyde. For lactic fermentation, there is no intermediate compound. It's formed. And in terms of the reversibility, alcoholic fermentation is irreversible. However, lactic fermentation is reversible, provided that oxygen is available. So this lactic acid can be converted back to pyruvate. When we compare about the ATP yield in aerobic and anaerobic respiration, aerobic respirations can generate an average of 32 ADP. However, anaerobic respiration can only generate a net gain of 2 ADP per glucose molecule. So during aerobic respiration, the complete oxidation of glucose molecule yield an average of 32 ADP. So this higher ADP yield is due to the electron transport chain as well as chemiosmosis during oxidative phosphorylation. Because oxygen acts as a finite electron acceptor in the electron transport chain, this allows ETC to function fully. However, in anaerobic respiration, there's no oxygen available to act as final electron acceptor. And because of this, electron transport chain cannot operate. So instead, fermentation now occurs. However, fermentation only involves glycolysis, which allow us to gain a net gain of 2 ADP per glucose molecule through substrate-linked phosphorylation. With that, we have come to the end of this lesson. I hope you have found this lesson helpful. If you have any questions, feel free to leave them in the comment section. See you in the next lesson. Have a great day.